The ocean is not flat. We are taught, in some early and persistent part of our minds, to think of it that way. A vast horizontal plane, its surface rippling under wind and weather, its floor somewhere far below in the dark. But the floor of the ocean is no more flat than the surface of a continent. It has mountains taller than any on land. It has plains extending for thousands of miles without a single feature. And it has places that plunge downward with such violence, such geological ferocity, that the human mind requires a moment to fully accept what the numbers mean. The Mariana Trench is one of those places. It sits in the western Pacific Ocean, roughly 200 kilometres east of the Mariana Islands, curving in a long crescent arc for about 2,550 kilometres, nearly the width of the continental United States, end to end. At its widest, it spans approximately 69 kilometres. At its deepest, it reaches a point that sits nearly 11 kilometres below the surface of the sea. 11 kilometres. To put that number into the body rather than just the intellect, if you stood at sea level and began walking straight down, you would pass through nearly 11 times the height of the Empire State Building before touching the floor of the Challenger Deep. If you placed Mount Everest at the bottom of the Mariana Trench, its summit would still sit more than 2 kilometres underwater. The weight of all that water pressing down on the bottom of the trench is roughly 8 tons per square inch, over 1,000 times the atmospheric pressure we walk through every day without noticing. This is not a place designed for human life. And yet humans have gone there, a small number of them, across a span of decades, in vessels that required extraordinary engineering, extraordinary nerve, and in some cases a remarkable tolerance for the knowledge that the entire ocean was above you and nothing was below. Their stories are worth telling carefully, but they are not the whole story, and perhaps not even the most interesting part of it. The trench existed for a very long time, before any human being knew it was there. It will exist long after the last evidence of human civilization has dissolved into sediment. To understand it fully, to sit with it, let it settle into the imagination, and feel the weight of what it actually is, you have to begin at the beginning, not with the explorers, with the rock. About 180 million years ago, give or take a few million on either side, a section of oceanic crust was forming at a spreading ridge somewhere in the central Pacific Ocean. Molten rock was rising from the mantle, cooling at the surface, and spreading outward in both directions from the ridge, the way lava flows from a crack rather than a cone, quietly and without drama. The resulting crust was thin, dense, and dark. Basaltic rock mostly. Nothing spectacular to look at. Just the routine business of a planet continuously building and rebuilding itself. That crust, over the following 180 million years, moved. Tectonic plates move slowly, a few centimetres per year, roughly the speed at which a human fingernail grows. But over the scale of deep geological time, that adds up. This particular section of crust, riding on the enormous Pacific plate, was carried steadily westward and northward across the ocean basin. As it moved, it cooled further, it contracted, it became denser and heavier, and at some point it reached the edge of the Mariana Plate, a smaller, younger piece of oceanic crust sitting to the west. What happened next is called subduction, and subduction is, in the simplest possible terms, one tectonic plate diving beneath another. The Pacific Plate, old and cold and heavy, began to angle downward beneath the lighter Mariana Plate. It did not do this dramatically or with visible force. The event unfolded across millions of years, at speeds that would make a glacier seem impatient, but the result was decisive. Where the Pacific Plate began to bend and dive, the ocean floor was pulled downward with it, and at the outer edge of the subduction zone, where the bending plate had not yet plunged fully into the mantle, a depression formed, a trench. The Mariana Trench is deep for several reasons that compound each other in a way that no other trench on Earth quite replicates. The Pacific Plate in this region is among the oldest oceanic crust on the planet, approximately 180 million years old. Old oceanic crust is cold. Cold rock is dense. Dense rock sinks more readily. The steeper the angle of subduction, the deeper the trench carved at the surface. Additionally, the trench lies far from any major continental landmass. In many other deep trenches around the world, Rivers carry enormous volumes of sediment into the sea, and that sediment gradually fills the trench from above, reducing its effective depth. The Mariana Trench has no such supply. It is remote, sediment-poor, and essentially self-cleaning. And finally, nearby fault lines cut through the Pacific Plate in a way that allows it to bend more steeply downward than it otherwise would, steepening the descent further. These three factors together, extreme age and density, sediment starvation, and a favourable fault geometry, conspire to produce a trench that is not just deep, but uniquely, almost structurally destined to be the deepest point on Earth. The trench is not static. The Pacific Plate is still moving. The subduction is ongoing. There are portions of the region where the plate descends at speeds that, by geological standards, are quite fast, and this creates seismic instability. The Mariana Trench sits within the broader Pacific Ring of Fire, the horseshoe-shaped zone of intense earthquake and volcanic activity that encircles the Pacific Basin. The neighbouring Mariana Islands to the west were built by the same subduction process. Magma from the descending Pacific Plate, melted by the heat and pressure of the mantle, rising through the overlying Mariana Plate and erupting at the surface. Every volcanic island in that chain is, in a sense, a byproduct of the same process that created the deepest point on Earth. The floor of the trench is, in all practical terms, a glimpse into the interior of the planet. The rocks exposed along its walls include some of the oldest oceanic crust anywhere on Earth. Pieces of the mantle itself have been lifted to near-surface levels through a process called serpentinization. Seawater interacting with deep mantle rock, chemically transforming it and producing hydrogen and methane gases in the process. Along the Mariana Forearc, in the shallower regions to the west of the trench's deepest portions, there are at least 10 active mud volcanoes. Features found nowhere else on Earth in quite the same form. Erupting slow slurries of serpentinite mud mixed with fragments of subducted seamounts, ancient coral reefs, and deep mantle material. One of those mud volcanoes, the South Chamorro Seamount, expels fluid with a pH approaching 12, as alkaline as household bleach. Another nearby site, the Champagne vent at the Ifuku submarine volcano, produces almost pure liquid carbon dioxide, a phenomenon observed in only one other location on Earth. And at the Daikoku submarine volcano, there is a convecting pool of liquid sulfur roiling at temperatures above the boiling point of water under the pressure of the depths. The only other confirmed location of a liquid sulfur pool is on Io, a volcanic moon of Jupiter. The Mariana Trench is not simply a hole. It is an active, complex, chemically extraordinary geological system, and we have barely begun to understand it. The trench was not known to exist until 1875. This is worth sitting with. For the entirety of human history before that year, the rise and fall of every civilization, every ocean voyage, every navigational achievement, the deepest point on Earth was simply unknown. Not just unexplored, unknown. Nobody suspected it was there. On the 23rd of March 1875, a British Royal Navy corvette named HMS Challenger was sailing through the Western Pacific as part of a 4-year scientific expedition that had departed Portsmouth in December 1872. The Challenger had been stripped of most of its guns and refitted with laboratories, sampling equipment, and enormous spools of sounding rope. The expedition's purpose was systematic: to take depth measurements, collect water samples, dredge the seafloor, and attempt to answer whether life could survive in the deep ocean at all. The Challenger had been doing this work for years by the time it reached the Western Pacific. The crew had become practiced at the routines of sounding, lowering weighted ropes into the water, waiting, feeling for the resistance of the seafloor. They did it at hundreds of stations across multiple oceans. Most of the time, the measurements came back within an expected range. At station number 225, they lowered the rope and it kept going. The sounding recorded a depth of 4,475 fathoms, approximately 8,184 meters. It was the deepest reading anyone had ever obtained from the ocean, and it was confirmed with a second sounding at the same location. The area was named the Swire Deep, after the expedition's navigating sub-lieutenant Herbert Swire. Later expeditions would reveal the full extent of what lay beneath, and the feature was eventually renamed Challenger Deep, in honor of the ship that first found it. It is worth noting that the Challenger's crew had no way of knowing what they had discovered. They recorded the measurement, confirmed it, and moved on. The full significance of that station, the realization that this was not merely a local depth anomaly, but the lowest point on the entire surface of the planet, would only emerge much later as subsequent surveys mapped the surrounding region and the shape of the trench became clear. The expedition returned to England in May 1876, having travelled nearly 70,000 nautical miles and collected data from 354 stations. The findings filled 50 published volumes, totaling 29,500 pages. Nearly 80% of all animal specimens collected were new to science. It is, without exaggeration, considered the founding voyage of modern oceanography. In 1951, 76 years after the original sounding, a British naval survey vessel called HMS Challenger 2, named deliberately in honour of its predecessor, returned to the area and used an echo sounder to take more precise measurements. The depth it recorded was substantially greater than what the original Challenger had found, in the region of 10,863 metres. The Challenger 2 had confirmed that this was not just deep. It was in a different category from every other place on Earth. The deepest point of the Mariana Trench was formally named Challenger Deep, carrying the names of both ships into the geographical record. Scientists were now certain that a place nearly 11 kilometres below sea level existed. Nobody had yet gone there. Nobody, for that matter, had gone anywhere close. The question of how to get there was not trivial. To understand what a descent to Challenger Deep actually requires, it helps to understand what depth does to pressure. At the surface of the ocean, atmospheric pressure is approximately 1 atmosphere. The weight of all the air above you, measured at sea level. For every 10 metres of depth in the ocean, pressure increases by approximately 1 additional atmosphere. At 100 metres deep, a depth reachable by some skilled free divers, the pressure is already 11 atmospheres. At 1,000 metres, 101 atmospheres. At the bottom of Challenger Deep, approximately 10,935 metres below the surface, the pressure reaches roughly 1,071 atmospheres, or approximately 8 tons per square inch. This is not a force that the human body can survive without engineering. It is not a force that most materials can survive without engineering. Water itself does not compress significantly at these pressures. It remains liquid, but gas does. Any air cavity in a vessel, any void, any space where matter is less dense than the surrounding water, will be crushed inward by the ocean pressing from every direction simultaneously. To dive to Challenger Deep in a conventional submarine would be to implode it. The solution, developed over several decades in the mid-20th century, was the bathyscaphe. A vessel designed not around rigidity alone, but around buoyancy management. The idea came from a Swiss physicist named Auguste Piccard, who had already set high-altitude balloon records in the stratosphere and saw the engineering principles as analogous. In both cases, the challenge was survival in an environment of extreme pressure differential. In both cases, the solution involved a heavy sphere to house the occupants, combined with a buoyancy system that could be adjusted to control depth. Piccard designed a bathyscaphe in which the crew sphere, a thick-walled steel globe just large enough for 2 people to sit, was suspended beneath a large tank filled with gasoline. Gasoline is less dense than seawater, so it provides positive buoyancy. To descend, ballast was released or flooding tanks opened to add weight. To ascend, ballast was dropped and the gasoline did the rest, lifting the vessel back toward the surface. It was his son, Jacques Piccard, who would take the design to its ultimate test. On the 23rd of January, 1960, Jacques Piccard and United States Navy Lieutenant Don Walsh climbed into the sphere of a bathyscape called the Trieste and began to descend. The weather above was not good. The seas were running heavy, with swells of about 7.5 metres. The Trieste pitched and rolled in the wave action until it was deep enough for the ocean to become calm around it. Then, the 2 men were alone in the dark. The descent took 4 hours and 47 minutes. They fell at roughly 1 metre per second, passing through the successive thermal layers of the ocean. The warm, sunlit surface water, the cold mid-water where temperatures drop sharply, the constant, near-freezing deep, The sphere measured about 2 meters in diameter. They had instruments and a small porthole, but little else. The world outside was complete darkness. At approximately 9,000 meters, the sphere cracked. Not catastrophically. A loud crack. Walsh described it as heard and felt simultaneously. Turned out to be one of the outer plexiglass window panels fracturing under the pressure. The inner hull, the steel sphere that actually contained them, was undamaged. They checked the instruments. The crack had not reached anything critical. They continued down. At 1 hour and 8 minutes past 8 in the morning, Guam time, the Trieste settled onto the floor of Challenger Deep in a cloud of pale diatomaceous ooze. The compressed, powdered remains of microscopic algae that had been settling down through the water column for millions of years. The ooze billowed up and filled their viewport. For the entire 20 minutes they spent on the bottom, they could see nothing through it. Walsh later recounted this with characteristic directness. They had come to the bottom of the world and could not see a thing. The dust never cleared. There was one observation from that brief visit that has since become famous and disputed in roughly equal measure. Piccard reported seeing what appeared to be a flatfish, something resembling a small sole, resting on the sediment just before the landing cloud obscured the viewport. Walsh was less certain. Decades later, he acknowledged that neither man was a trained marine biologist, and that the animal could have been something else, perhaps a large sea cucumber, which was a known resident of extreme depths. Scientists who have since studied the physiological limits of fish, the maximum depths at which bone-bearing vertebrates can survive, given the biochemical constraints of maintaining protein function under pressure, have generally concluded that a true flatfish at Challenger Deep is biologically impossible. The disputed fish remains unresolved. Neither man could prove what they saw. No photograph exists. The Trieste carried no camera capable of capturing an image through the cloud-filled viewport. It is one of the first of many things about Challenger Deep that turns out to be genuinely, permanently uncertain. Walsh and Piccard had spent 20 minutes at the bottom of the world. They ascended in 3 hours and 15 minutes. When they surfaced and climbed onto the deck of the Trieste, they expected a prompt return by other expeditions within a few years. Walsh estimated 2 or 3 years. It would be 52 years before any human being returned. The gap between 1960 and 2012 is one of the stranger facts in the history of exploration. To reach the highest point on Earth, the summit of Mount Everest, first climbed in 1953, 7 years before the Trieste dive. More than 9,000 people have since made the ascent. To reach the lowest, only a handful of humans have ever gone. And in the decades following Walsh and Piccard's descent, the United States Navy lost interest in manned deep ocean exploration, and the Trieste program was eventually folded. No nation, no institution, no private individual made the attempt for over half a century. This is, I think, a genuinely revealing fact about human priorities. We built the equipment and the knowledge to reach the deepest point on Earth, and then we collectively decided there were other things to do. The return came in March 2012, when filmmaker James Cameron, not a scientist, but a man of extraordinary technical seriousness and deep personal obsession with the ocean floor, descended in a submersible he had spent 7 years designing and building. The deep-sea Challenger was 24 feet long and oriented vertically, designed to fall like a torpedo rather than float horizontally like a conventional submersible. Cameron reached the bottom alone on the 26th of March 2012, touching down at a depth of 10,908 meters. He spent 3 hours on the bottom. Unlike Walsh and Piccard, he could see. The sediment cloud cleared. What he saw, he later described as a featureless plain, extending to the horizon in every direction. Soft, nearly flat, pale, and quiet. A lunar landscape was the comparison he reached for more than once. No hydrothermal vents visible from this position. No obvious clusters of complex life. Just the floor of the world, immense and still. Cameron collected sediment and water samples, and the scientific analysis of his mission would later confirm the presence of microbial communities in the sediment, populations of bacteria and archaea that had adapted to the crushing pressure and near-freezing temperatures, living not on photosynthesis or the trickle of organic material from above, but on chemical energy extracted from the rock beneath them. The next significant manned visit came in 2019, when a former private equity investor and retired naval officer named Vescovo descended to 10,927 metres, about 16 metres deeper than Cameron's record, as part of a self-funded mission to reach the deepest point in each of the world's 5 major oceans. Vescovo made multiple dives to Challenger Deep over several days. His submersible, the limiting factor, was more capable than Cameron's in terms of time on the bottom, and the scientific team collected additional biological and geological specimens. Vescovo reported 2 things about his visit that deserve to be taken together. The first was scientific: colourful rocky outcrops on the walls of the trench that appeared to be chemical deposits, bottom-dwelling holothurians moving across the sediment, and amphipods, small crustacean scavengers in surprising abundance. The second was not scientific in any technical sense, but it carried a weight that has not diminished since. At the deepest point on Earth, Vescovo found a plastic bag and several candy wrappers. There is something in that image that resists easy processing. Here is the most remote, inaccessible, extreme environment on the planet. Here is a place that requires millions of dollars of engineering and years of preparation to reach. And settled on the floor of it, carried there by ocean currents no one fully understands, from a surface source no one has been able to identify with certainty, was a piece of ordinary human refuse, the kind of thing that blows off a deck or falls off a pier, or gets dropped in a river on the far side of the world. In 2024, a beer bottle was photographed on the floor of Challenger Deep's western pool during a subsequent dive. This too has a weight to it that goes beyond the object itself. It is not that the presence of litter at the deepest point on Earth is surprising, given what we know about ocean circulation. It is that we now know it, and we have the image, and the image refuses to be unfelt. To understand why anything lives at the bottom of the Mariana Trench, you first need to understand why it should not. The challenges to life in the hadal zone, the technical term for ocean depths below 6,000 metres, are severe and stacked upon each other in ways that no surface environment quite replicates. The pressure at Challenger Deep, as already noted, is over 1,000 atmospheres. The temperature hovers between 1 and 4 degrees Celsius. There is no light of any kind. Photosynthesis, the process that drives the energy web of nearly every ecosystem on Earth, is entirely absent. The supply of food is tenuous. Organic material, bacteria, the bodies of dead organisms from far above, sinking slowly through kilometres of water before reaching the bottom. And yet, life is there, not merely present in trace amounts, clinging to survival at the edge of the possible. Life at the bottom of the Mariana Trench is, by the standards of extreme environments, relatively abundant and surprisingly diverse. The reason begins with pressure and with a molecule that took scientists several decades to fully understand. Every cell in every living thing contains proteins, complex molecules folded into precise 3-dimensional shapes, whose function depends entirely on maintaining that shape. Pressure disrupts protein folding. At the pressures found at Challenger Deep, the physical forces acting on the water molecules inside a cell begin to distort those molecules away from the configurations they need to adopt. Proteins unfold, enzymes stop working, cell membranes lose their proper structure. The organism, at a biochemical level, falls apart. Marine fish, like most vertebrates, are particularly vulnerable to this. They carry gas in their swim bladders. Their proteins fold in configurations that assume surface pressure water chemistry. They cannot simply descend into the hadal zone and keep functioning, but the fish and the amphipods and the bacteria and the archaea that actually live in the hadal zone have found a chemical solution. Inside their cells, they accumulate a compound called trimethylamine N oxide, or TMAO. This molecule acts as what scientists call a piezolyte, a pressure solute. It works by interacting with the water molecules in the cell, encouraging them to maintain the hydrogen bonding configurations that allow proteins to fold correctly even under extreme pressure. TMAO, in effect, counteracts the distorting influence of pressure on the cell's interior water chemistry. What makes this especially striking is that the concentration of TMAO in fish tissue increases predictably with depth. Shallow water fish have relatively little of it. Deep-sea fish have more. Hadal fish have the highest concentrations recorded. It is a literal biochemical signature of depth, a measurable adaptation that scales with the challenge. Research published by Paul Yancey at Whitman College and colleagues, using specimens from the Comedic and Mariana Trenches, demonstrated that the correlation holds across species and across depth zones with a regularity that leaves little room for doubt. There is a hard limit to this, however. TMAO is an osmolyte. It draws water into the cell, and at some point, the concentration required to counteract pressure becomes so high that it creates a different kind of physiological problem. Scientists have calculated that this limit corresponds to a maximum depth of approximately 8,200 meters for bony fish. Below that depth, the TMAO required to survive pressure would itself become toxic in its osmotic effects. This is why no bony fish has ever been reliably confirmed below about 8,400 meters. The deepest fish ever recorded, a species of hadal snailfish, Pseudoliparis swire, named for the Challenger's sub-lieutenant Herbert Swire, was filmed at a depth of approximately 8,300 meters in the Mariana Trench. It is a translucent, gelatinous-looking creature with an oddly domestic appearance, pale, soft-bodied, moving through the dark water with slow, deliberate fins. Its bones are reduced and incompletely mineralized, almost cartilaginous, likely as an adaptation to avoid rigid structures that would be vulnerable to pressure damage. Its genome, sequenced by Chinese researchers and published in the journal Nature Ecology and Evolution in 2019, contains specific mutations in the gene that controls bone mineralization and shows expanded copies of the enzyme responsible for TMAO production. Evolution does not intend, but if it did, you could almost say that the Mariana snailfish was designed for this place. Below 8,400 meters, no vertebrate fish has been found. But this does not mean the trench below that depth is lifeless. It means the life there is different in kind. The dominant animals at the very deepest points of the trench are amphipods, small crustaceans that resemble oversized shrimp, related to the sandhoppers found in tide pools, but vastly different in their adaptations. Some amphipod species at hadal depths can reach surprising sizes, with supergiant individuals documented at over 25 centimeters in length. They are scavengers, drawn to food falls, the bodies of larger organisms that sink from above, occasionally including whale carcasses, seabirds, or fish. When food arrives at the bottom, amphipods can aggregate in extraordinary densities around it, their numbers sometimes in the thousands. Amphipods from the Mariana Trench carry their own chemical adaptations. A 2025 study in Cell, part of the Chinese Mariana Trench Environment and Ecology Research Project, found that hadal amphipod populations have developed symbiotic relationships with specific gut microorganisms that enhance their pressure tolerance and their ability to metabolize the sparse chemical energy available at depth. The amphipods also carry TMAO and additional pressure counteractant compounds, including skilo-inositol, which is, separately, being studied as a potential treatment for Alzheimer's disease. The deep ocean offers unexpected pharmaceutical implications, and this is one of them. The floor of the trench, below the snailfish depth limit, in the last few kilometers of depth, is inhabited primarily by invertebrates and single-celled organisms. Sea cucumbers, or holothurians, move slowly across the sediment, ingesting organic material. Polychaete worms and foraminifera occupy the sediment itself. And there are the xenophyophores, among the strangest organisms on Earth, and, in one sense, among the most impressive. Xenophyophores are single-celled organisms. This is worth repeating plainly. A single cell, and yet individual xenophyophores have been documented at sizes exceeding 10 centimeters, making them among the largest single-celled organisms known to exist anywhere on Earth. They are related to amoebas and foraminifera, and they build structures called tests from sediment particles and organic material, agglutinating the surrounding environment into elaborate shelters for their single nucleus and the complex cytoplasm that constitutes their entire body. They were found at depths of more than 10.5 kilometers in the Serena Deep of the Mariana Trench during a 2011 Scripps Institution of Oceanography expedition, the deepest confirmed presence of any such organism at the time. The sediment at Challenger Deep itself, the soft, pale ooze that Walsh and Piccard disturbed with their landing and that Cameron encountered on his dive, is alive in ways that are still being catalogued. The Mariana Trench Environment and Ecology Research Project, publishing in March 2025, identified over 6,000 new microbial species in sediment samples from the hadal zone, describing two specific biochemical adaptations, novel protein configurations and altered membrane lipid compositions that allow microorganisms to function at pressures that would destroy any surface-adapted cell. The food web at these depths is powered not by sunlight, but by two other sources. The first is marine snow, the continuous slow rain of organic particles from above. Dead plankton, fecal pellets, shed skin, the fragments of organisms that lived and died in the sunlit upper ocean, broken down progressively as they fall through 200, then 2,000, then 8,000 meters of water. By the time anything reaches the floor of Challenger Deep, it has been filtered through multiple layers of consumers and decomposers. What arrives is sparse, but the organisms that live at the bottom are extremely efficient at capturing it. The second energy source is chemosynthesis. In the absence of photosynthesis, certain bacteria and archaea can extract energy from chemical reactions, the oxidation of hydrogen sulfide, methane, or hydrogen gas produced by the geological processes of the trench itself, the serpentinization reactions that produce hydrogen deep in the mantle rock, the chemical venting from mud volcanoes and cold seeps. All of this provides a slow trickle of chemical energy to microbial communities that have no dependence on the surface world whatsoever. In those communities, there is something that I find genuinely moving and worth pausing on. The microbes at the floor of the Mariana Trench, the archaea and bacteria living in sediment that has never seen light, sustained by the chemistry of rock and water, adapted to pressures that would crush any surface organism, are not the simple, primitive life forms we might be tempted to imagine. They are the product of billions of years of evolution. Their biochemistry is extraordinarily sophisticated. They have solved problems that our own cells cannot solve. And they were here before the first fish appeared in the ocean above them, and before the first plant colonized the land, and before the first dinosaur breathed. They are older, in a meaningful evolutionary sense, than almost everything we typically call life. They simply live in a place we cannot. The question the Mariana Trench raises most persistently is not about the trench itself. It is about everywhere else. In our solar system, there are several worlds that scientists believe, with varying degrees of confidence, based on increasingly strong evidence, contain subsurface oceans of liquid water. Europa, a moon of Jupiter, is covered in a crust of ice that is estimated to overlay a global ocean roughly twice the volume of Earth's. Enceladus, a moon of Saturn, actively vents water vapor and ice particles from cracks near its south pole, providing direct evidence of liquid water beneath the surface. Both of these oceans exist in complete darkness, under enormous pressure, in environments where photosynthesis is impossible and the energy must come from chemistry or tidal heating or both. Before the deep ocean exploration of the 20th century, the scientific consensus was that life required sunlight as its primary energy source. This was not an unreasonable assumption. Every ecosystem on Earth that humans had studied was ultimately dependent on photosynthesis. Then, in 1977, a joint expedition by Woods Hole Oceanographic Institution and Scripps Institution of Oceanography sent a small submersible called Alvin to explore the Galapagos Rift, a section of the mid-ocean ridge system in the eastern Pacific, and found hydrothermal vents surrounded by dense communities of tube worms, clams, crabs, and fish, all supported by chemosynthetic bacteria with no dependence on sunlight whatsoever. It overturned one of the foundational assumptions of biology. The discovery in the subsequent decades of life at the very bottom of the Mariana Trench, in complete darkness, under pressures that would liquefy a human body, at temperatures near freezing, sustained by chemistry rather than light, extended that revolution further. Life, it turns out, does not require sunlight. It does not require moderate pressure. It does not require surface conditions of any kind. It requires liquid water, chemical energy, and time. Europa has liquid water. Enceladus has liquid water. Titan, a moon of Saturn, has liquid methane on its surface and may have a water ocean beneath its crust. These are not marginal candidates for life. They are, by the standards set by the Mariana Trench, plausible habitats. NASA astrobiologist Kevin Hand, who was part of the team that photographed what appeared to be microbial filaments on the floor of the Mariana Trench's Serena Deep during the 2012 Cameron expedition, put it directly: The existence of a chemosynthetic microbial ecosystem in the deepest, darkest, highest pressure region of Earth's ocean serves as a guide for the potential habitability of Europa's ocean. He was not speculating. He was drawing a comparison grounded in specific biochemical and physical parallels. I am not claiming that life exists on Europa. Nobody knows whether it does. But the Mariana Trench has done something significant to the terms of that conversation. Before we knew that life could thrive at 11 kilometres depth, in absolute darkness, under pressures exceeding 1,000 atmospheres, the question of life elsewhere in the solar system had a particular answer baked into its framing. Life needs what we see on Earth's surface. Now that framing has been revised, life may need only what the Mariana Trench provides: chemistry, liquid water, and time. The universe becomes, in some small but permanent way, a more hospitable place. In 2019, the year of Victor Vescovo's record-breaking dive to Challenger Deep, a study published in the journal Royal Society Open Science reported something that had been expected and dreaded in equal measure. Microplastics were found in the bodies of amphipods from the Mariana Trench. Not just a few individuals. Every single crustacean tested, from 6 different hadal trenches across the Pacific Rim, contained at least one piece of synthetic material. The average in the Mariana Trench population was the highest of all 6 trenches. One individual contained 8 separate microplastic particles. The scientists who published the study chose their words carefully. One of them, William Reed of Newcastle University, described it as probably the saddest piece of research he had ever been involved with. The microplastics included nylon fibers, polyethylene, polyamide, and rayon, the everyday materials of clothing, packaging, and industrial products. They had arrived at the floor of the trench through ocean circulation, settling through the water column from surface accumulations, concentrated by the funnel geometry of the V-shaped trench into densities that, in the bottom sediment, have been measured at between 202,200 particles per litre. For comparison, open ocean subsurface waters typically contain between 2 and 14 particles per litre. The hadal zone has been called the largest sink for microplastic debris on Earth. The same year, researchers from Shanghai Ocean University published a study in Scientific American documenting the presence of carbon-14, radioactive carbon produced by nuclear weapons testing, in the atmosphere during the 1950s and 60s in the flesh of amphipods from the Mariana Trench. The carbon had entered the food web at the surface, been incorporated into plankton and small organisms, and worked its way down through the food chain over decades until it reached the bottom of the trench. An Earth scientist involved in the study observed, "Typically we say the trenches are far away from us. They are very deep, and they are pristine. But actually they are not. Everything can get into the trenches." In 2016, researchers examining crustacean scavengers from depths between 7,800 and 10,250 metres within the Mariana Trench found extremely elevated concentrations of polychlorinated biphenyls, PCBs, in their tissues. PCBs are industrial chemical compounds banned in most countries by the late 1970s due to their extreme toxicity and persistence in the environment. They do not break down, they accumulate. In the flesh of animals living at the bottom of the Mariana Trench, scientists found PCB concentrations 50 times higher than those recorded in crabs from China's most heavily polluted river systems. It is not easy to hold this information calmly. It represents something significant about the relationship between industrial civilization and the planet it inhabits. We have contaminated the most inaccessible place on Earth, a place that required humanity's best engineering to reach, even briefly, with the routine byproducts of ordinary life. The plastic bag that someone threw away, the PCB that leached from electrical equipment, the radioactive carbon that fell from a bomb test in the Nevada desert. All of it finds its way, eventually, to the floor of the Mariana Trench. The trench is not pristine. It never will be again. This is perhaps the starkest statement that can be made about the reach of human impact on the natural world. There is one more thing about the Mariana Trench that deserves careful attention, and it concerns how much of it remains unknown. Despite the decades of expeditions, the multiple manned dives, the remotely operated vehicles, and autonomous landers and sediment samplers, what has actually been seen of the Mariana Trench directly observed by human eyes or by cameras, is a vanishingly small fraction of the total. A 2024 study published in the journal Science, led by researcher Katie Croft-Bell of the Ocean Discovery League, assembled a database of every deep-sea observation expedition since 1958, more than 43,000 dives in total, and calculated the cumulative area of seafloor that had actually been seen. The result was startling. Humans have directly observed approximately 0.001% of the deep seafloor, not 1%, 0.001%. The study noted that most of the expeditions were concentrated within 200 nautical miles of the coastlines of the United States, Japan, and New Zealand. Vast regions of the deep ocean, including much of the Indian Ocean, the deep South Atlantic, and enormous tracts of the Pacific, have essentially never been observed at any resolution. The Mariana Trench itself has received far more attention than most hadal features by virtue of its status as the deepest point on Earth. And even here, the picture is incomplete. The Challenger Deep alone consists of 3 distinct deep basins: the Eastern Pool, the Central Pool, and the Western Pool, each with slightly different depths and presumably different biological and geological characteristics. The Serena Deep, lying about 240 kilometres to the east, reaches nearly 10.5 kilometres depth and was only discovered decades after Challenger Deep was mapped. Further unexplored regions may exist within the broader Mariana Trench system. Meanwhile, the Seabed 2030 project, an international collaboration aiming to produce a complete map of the world's ocean floor by the year 2030, had mapped approximately 26% of the seafloor as of late 2024, up from 6% when the project launched in 2017. At the current rate of progress, the project would require several additional decades to complete its goal, even at accelerated effort. Much of the world's ocean floor has never been mapped to any modern resolution, let alone observed. This is worth saying slowly, because it is easy to underestimate. We live on a planet that is 70% ocean, and we have directly observed approximately 0.001% of its floor. We have better maps of the surface of Mars than of the bottom of our own sea. The Mariana Trench, the one place in the ocean that has received the most attention precisely because of its extraordinary depth, has been visited by humans fewer than a dozen times in total. 3 people have reached Challenger Deep for scientific purposes, and a small number of additional divers have descended as passengers in vessels funded partly by the proceeds of deep-sea tourism. The entire cumulative time that human beings have spent at the bottom of Challenger Deep, across all expeditions since 1960, amounts to a few tens of hours. This is not a complaint. It is simply the reality of what the deep ocean is and what it costs to access it. But it shapes what we know and what we don't. The 6,000 new microbial species identified in the 2025 TMAO project study were found in samples collected from a comparatively tiny area of the hadal zone. The discoveries of giant xenophyophores, of novel snailfish species, of chemosynthetic microbial communities on rock outcrops, each of these came from a handful of dives to a handful of locations within a trench that is 2,500 kilometres long and 69 kilometres wide. What lives in the unexplored remainder is, quite genuinely, unknown. Scientists estimate that a minimum of 2 million marine species remain undiscovered. Many of them live in the deep ocean. A significant fraction almost certainly lives in hadal trenches. Some may live only in the Mariana Trench, in precisely the kinds of isolated, pressure-adapted, chemosynthetically sustained communities that have only been glimpsed at the very few points humans have reached. There is a reasonable case that the most biologically interesting environments on Earth are also the least explored. The hadal zone covers a small percentage of total ocean area, but represents a unique suite of conditions: extreme pressure, isolation, geological activity, chemical energy sources, that favour evolutionary novelty. Every time a well-equipped expedition has gone to a new hadal location, it has found species unknown to science. This is not coincidence. It is what happens when biology is given extreme conditions and a very long time. Consider what the planet was doing while the Pacific Plate was forming and slowly crossing the ocean toward the subduction zone that became the Mariana Trench. 180 million years ago, dinosaurs were new. The first mammals were small nocturnal creatures barely larger than shrews. The Atlantic Ocean did not yet exist. The continents that would become North America, Europe, Africa, and South America were still partially joined in configurations that would be unrecognizable to any modern eye. The world had no grass, no flowers, no birds, no whales. The rock at the western edge of the Pacific Plate, the rock that is now bending downward into the mantle beneath the Mariana Islands, the rock whose subduction created the Mariana Trench, was already formed by then. It was already on the ocean floor, cooling and densifying, beginning the slow migration that would carry it to the subduction zone over the following tens of millions of years. It has been descending beneath the Mariana Plate for approximately 40 million years. It is now perhaps 300 kilometres into the mantle in some portions of the subducting slab, far below any direct human observation, unreachable by any drilling technology we possess. At those depths, the pressure and temperature transform the rock completely, releasing water and other volatiles that rise into the overlying mantle, lowering its melting point, producing the magmas that feed the volcanic islands above. The Mariana Trench is not just the deepest place on Earth. It is one of the places where the planet actively recycles its own crust, where old ocean floor that was born at a spreading ridge and spent 180 million years crossing the Pacific returns to the mantle from which it came. The trench is a seam in the fabric of the planet, the point where one geological era ends and new material is born. The sediment that falls through the water column and settles at the floor of Challenger Deep, the diatomaceous ooze, the marine snow, the organic particles from the world above, is eventually incorporated into the descending slab and carried down with it. In geological terms, everything that reaches the bottom of the Mariana Trench is on its way somewhere else. The trench is a way station between the ocean surface and the deep interior of the Earth. This is what it means that the deepest point on the planet's surface is also one of the most geologically active. The trench exists because the planet is still in motion, still cycling its crust, still driving the conveyor belt of subduction that has regulated the composition of the oceans and atmosphere for hundreds of millions of years. Without subduction, the planet's surface chemistry would be different. The carbon cycle, the water cycle, the volcanic systems that produced the conditions under which life first appeared, all of these depend, in complex and not fully understood ways, on the return of oceanic crust into the mantle. The Mariana Trench is not an anomaly. It is not a wound or a scar. It is the planet working as intended. What does it mean, standing on the surface of the Earth, above 2 kilometres of atmosphere, above 11 kilometres of ocean, above the invisible floor of the world, to know that this place exists? I find myself resistant to the language of awe that tends to gather around the Mariana Trench in popular descriptions. It is a word that creates distance where I think the point is proximity. The trench is not awesome in the sense of remote and incomprehensible. It is astonishing in the sense of being exactly what it is. A piece of the planet governed by the same physics as everything else, obeying the same chemistry, housing the same kind of life that has always found its way into every available space. What makes it remarkable is not that it breaks the rules, it is that it follows them so completely at such an extreme. Pressure obeys an equation. You can calculate it. Temperature in the deep ocean follows gradients that have been mapped. The chemistry of TMAO adaptation follows a gradient so predictable that you can use it to estimate an organism's depth range from a tissue sample. The geology of the trench follows from plate tectonics that operate identically everywhere on Earth. Everything about the Mariana Trench is, in principle, explicable. And yet the sum of those explanations is a place where single-celled organisms 4 inches wide build themselves castles out of sediment, where translucent fish evolved to fill their tissues with a compound that reorders the physics of water, where an 18-year-old plate of rock is being returned to the mantle after a crossing that began before the first bird flew. The universe does not need to be strange to be extraordinary. The Mariana Trench is exactly what physics and chemistry and deep time produce when left to themselves for long enough. It is not a special exception. It is the planet being the planet. That is, I think, the most affecting thing about it. At the bottom of Challenger Deep, the water temperature is between 1 and 4 degrees Celsius. It has probably been in that range for millions of years, varying only with the slow changes of ocean circulation as continents moved and closed and opened ocean passages over geological time. The organisms that live there have no experience of any other temperature. They have no concept of sunlight, no awareness of seasons, no knowledge of the surface world that is 11 kilometres above them. They have been there, or their ancestors have been there, for an almost incalculable span of time. Their lineages predate every feature of the world that we would find recognisable. They were processing chemical energy in the dark while the first fish were learning to breathe air and climb out of shallow seas onto land. And they are still there, right now, in the dark and the cold and the tremendous quiet, doing what they have always done. There is something settling in that thought. Not melancholy, not disturbing, but genuinely settling. The deepest place on Earth has been occupied for longer than we can meaningfully imagine, by life that requires nothing from the surface world, and will continue long after the surface world has changed beyond recognition. The trench is patient in the way that geology is patient. It operates on timescales that make human history look like the first second of a very long film. Let that descend with you now, as you rest. The ocean is 11 kilometres deep in its deepest place. The water is cold and still and absolute. Life is there, has always been there, carrying on in the dark with a kind of untroubled persistence that is, in the end, more reassuring than any other fact I know. The deepest place on Earth is not empty. It never was. Before the floor, there is the descent, and the descent through 11 kilometres of ocean is itself a journey through multiple, entirely distinct worlds, each one layered above the last, each one governed by its own conditions of light and temperature and pressure and life. Start at the surface. The sunlit zone, scientists call it the epipelagic zone, from the Greek for upon the sea, extends from the surface down to roughly 200 metres. This is the ocean most of us know, or think we know. It is lit, warm by ocean standards, and driven by photosynthesis. Phytoplankton, microscopic plant-like organisms that form the base of the ocean food web, capture sunlight and convert it into organic carbon through the same biochemical process that land plants use. The entire productivity of the upper ocean flows from this. Every fish, every whale, every seabird ultimately depends on what happens in these top 200 metres. The sunlit zone is turbulent, dynamic, and in many regions intensely productive. But it represents only about 2% of the total ocean volume. Below it, the ocean is dark. The mesopelagic zone, from approximately 200 to 1,000 metres, is sometimes called the twilight zone. Sunlight reaches it only in attenuated, filtered form, enough for some organisms to detect and navigate by, but not enough to power photosynthesis. This zone is inhabited by organisms that migrate vertically on a daily cycle, rising to feed in the productive surface waters at night and retreating to the dark during daylight hours to avoid predators. The daily vertical migration of mesopelagic organisms is the largest animal movement on Earth. Billions of small fish, crustaceans and squid ascending and descending each day across hundreds of metres, carrying nutrients from deep water up toward the surface and then returning. The bathypelagic zone runs from 1,000 to 4,000 metres. Here, there is no light at all. The temperature is uniformly cold, between 2 and 4 degrees Celsius throughout. The pressure has increased to between 100 and 400 atmospheres. Life continues, but it is sparse compared to the zones above. The organisms here, various fish, crustaceans, jellyfish, cephalopods, are adapted to a world of complete darkness and sporadic food. Many have evolved bioluminescence, producing their own light through chemical reactions within their tissues, used for communication, predation, and evasion. Below 4,000 metres, the abyssal zone extends to the edge of the hadal features, the trenches. This is the largest volume of any ocean zone, the vast cold plains that constitute most of the deep seafloor. Temperature is near freezing, pressure is 400 to 1,000 atmospheres. Food is scarce. The organisms here are slow-moving, metabolically conservative, and often long-lived. Sea cucumbers, brittle stars, polychaete worms, and various species of fish and crustaceans inhabit the abyssal plains in densities that are low by surface standards, but distributed over an enormous area. And then the trench. Below 6,000 meters, the hadal zone, named for Hades, the Greek underworld, though the naming is perhaps more dramatic than the science strictly requires. The hadal zone includes all the deep ocean trenches, the Mariana, the Tonga, the Philippine, the Kermadec, and others. Each is a separate, isolated environment, a pocket of extreme depth separated from other hadal features by thousands of meters of shallower abyssal plain. The organisms in each trench have been isolated from those in other trenches long enough for evolution to have produced distinct populations and, in some cases, distinct species. This isolation is significant. The Mariana Trench is not a portal to some unified deep world shared with the Philippine Trench or the Japan Trench. It is, biologically speaking, an island, a fragment of extreme habitat surrounded by the shallower abyssal plain in the same way that a volcanic island is surrounded by ocean. Species that cannot survive at depths shallower than 6,000 meters are restricted to their trench. They cannot migrate between trenches except by ascending, which many are biochemically adapted against. Recent genetic studies of hadal amphipods have confirmed this pattern. The amphipod population of the Mariana Trench's western basin shows clear genetic divergence from populations in the Yap Trench and the West Philippine Basin, separated by shallower geography that acts as an impassable barrier. Within the Mariana Trench itself, depth gradient studies have found no significant population differentiation. The amphipods can tolerate pressure across the full range of trench depths, but the horizontal isolation between trenches is real and measurable in the genome. Evolution in the hadal zone has been working in isolated pockets. Each trench is its own experiment. To live at the bottom of the Mariana Trench is to depend on the surface world in a way that is easy to overlook. The chemosynthetic microbes at the very bottom can sustain themselves on chemical energy from the rock, but the larger animals, the amphipods, the holothurians, the occasional polychaete worm, depend on organic carbon that arrives from above. And the mechanism of that arrival is one of the stranger features of the deep ocean. Marine snow is the informal name for the continuous, slow precipitation of organic particles from the upper ocean toward the seafloor. It consists of dead phytoplankton and zooplankton, fragments of larger organisms, bacterial aggregates bound together by organic mucus into loose flocks, fecal pellets from crustaceans and fish, shed mucus from appendicularians, small filter-feeding invertebrates that build elaborate houses of mucus and then discard them when they become clogged, and bits of material from essentially every biological process occurring in the sunlit zone. As this material falls through the water column, it is consumed, fragmented, and repackaged at multiple levels. Bacteria colonize the falling particles and begin breaking them down. Midwater organisms intercept larger pieces, consume them, and produce smaller fragments and new fecal pellets that continue the descent. What reaches the abyssal zone after passing through 4 kilometers of this biological gauntlet is heavily processed, smaller, less nutritious per particle than when it started, and present in quantities that represent perhaps 1 to 2% of the original surface productivity. What reaches the floor of the Mariana Trench, 11 kilometers below the sunlit zone, is a fraction of a fraction. The food supply is extraordinarily sparse. Organisms at these depths have adapted to this reality in multiple ways. Their metabolic rates are extremely low. They require far less energy per unit of body mass than equivalent animals at the surface. Their growth rates are correspondingly slow. Some deep-sea invertebrates are believed to live for centuries precisely because they grow so slowly and expend so little energy that their biochemical machinery degrades at a drastically reduced rate. There is also a concentration mechanism specific to the trench itself. The funnel geometry of the V-shaped trench, wide at the top, narrowing toward the bottom, acts as a sediment trap. Marine snow and organic particles that settle through the water column anywhere within the horizontal extent of the trench are gradually channeled toward the deepest point by the trench's slope. The floor of Challenger Deep likely receives somewhat more organic material per unit area than a comparably deep location on the open abyssal plain, precisely because the trench geometry concentrates what falls into it. This concentration effect may partly explain why amphipod populations at the deepest points of the trench can be so surprisingly dense when food arrives. The trench is, in a sense, a natural collecting funnel, and for scavengers adapted to exploit food falls, the accumulated organic matter at the bottom represents a predictable, if infrequent, resource. The history of how humans have understood the deep ocean is a history of being wrong, and then being corrected, and then being wrong in a new direction. Before the 19th century, the question of life in the deep ocean was barely even asked. The ocean was a medium of transport, a source of food near the surface, a dangerous environment to be navigated rather than explored. Its floor was, for most of recorded history, simply unimaginable. The first serious scientific hypothesis about the deep ocean floor was proposed by Edward Forbes, a British naturalist who conducted dredging expeditions in the Aegean Sea in the 1840s. Forbes noticed that as he dredged progressively deeper, from shallow coastal waters down to several hundred meters, the diversity and abundance of animals decreased systematically. Extrapolating from this trend, he proposed what became known as the azoic hypothesis, the idea that below, approximately 550 meters, the ocean floor was completely devoid of life, too cold, too dark, too compressed. The azoic hypothesis was influential. It was also wrong in almost every particular. Even at the time, there were dissenting voices. Sir James Ross, the polar explorer, regularly pulled up living animals from sounding lines at depths exceeding 550 meters. In 1862, telegraph cable workers repairing a broken undersea line in the North Atlantic found living organisms adhering to the cable from depths far greater than Forbes's proposed limit. In 1866, a Norwegian naturalist named Georg Ossian Sars dredged a stalked crinoid, a lily-shaped echinoderm related to sea urchins, from nearly 500 meters off the Norwegian coast. The animal was a species from a group thought to be long extinct. The implication was extraordinary. The deep ocean might be a refuge, a place where ancient lineages persisted long after they had vanished from the shallower, more competitive environments above. These findings pushed the Royal Society of London to advocate for a systematic expedition to test the azoic hypothesis directly. The result was the Challenger expedition of 1872 to 1876. The Challenger settled the question decisively. At every depth it sampled, including the very deepest measurements it could achieve with its equipment, it found life. The deep ocean was not empty. It was inhabited, differently than the surface, more sparsely, with different communities adapted to different conditions, but inhabited at every depth. Forbes had extrapolated correctly within the narrow range of his data, and catastrophically beyond it. The lesson was not simply that Forbes was wrong. The lesson was that the absence of evidence in a place extremely difficult to access is a very poor indicator of actual absence. When the technology for observation improved, the answers changed. This lesson has been relearned many times since. Each major improvement in deep ocean observational technology, from the Challenger's dredges to the Trieste's porthole to modern remotely operated vehicles with high-definition cameras, has produced discoveries that were not predicted and often not imagined. Every time someone has looked carefully at a new deep ocean location, they have found things that were not expected. Between the Trieste's 20-minute visit in 1960 and James Cameron's return in 2012, the Challenger Deep was not entirely unvisited by machines. In 1995, a Japanese remotely operated vehicle called the Kaiko descended to 10,911 meters, the deepest dive of any unmanned vehicle to that date, and for the first time took photographs and collected sediment samples from the floor of the trench. The Kaiko was operated by the Japan Agency for Marine Earth Science and Technology, known as JAMSTEC, and it represented the first genuine scientific survey of the Challenger deep floor, where the Trieste had been limited by its cloud of disturbed sediment and its 20-minute bottom time. The Kaiko could hover, maneuver, and film. Its cameras showed a featureless plain, as Walsh and Piccard had described. Soft sediment extending in every direction, pale in the vehicle's lights, utterly still. The sediment samples Kaiko retrieved changed the understanding of life at the deepest point on Earth. Analysis revealed hundreds of species of microorganisms living in the sediment, bacteria, archaea, foraminifera, in densities far above zero. The deep-sea biologist Lisa Levin, reviewing the Kaiko findings, described Challenger Deep as surprisingly alive for a place of such extreme conditions. The Kaiko made multiple dives over several years, building the first detailed picture of Challenger Deep ecology. In 2003, the Kaiko's primary vehicle was lost at sea when its tether snapped during a typhoon near Shikoku. Japan subsequently developed a replacement vehicle, but the loss was a significant setback for hadal research. It reflects something worth noting. The equipment required to reach Challenger Deep is expensive, fragile, and exposed to mechanical stresses that test the limits of engineering. The barriers to deep ocean research are not primarily intellectual or motivational. They are physical and financial, and the ocean does not make them easy. Since Vescovo's dives in 2019, additional visits to Challenger Deep have been made by Chinese manned submersibles. In November 2020, China's Fendosia, the name means striver, carried scientists to depths exceeding 10,900 meters on multiple dives. And the biological and geological data collected as part of the MIA project represents the most systematic scientific program to date at hadal depths. The 2025 Cell paper emerging from this program, documenting over 6,000 new microbial species and describing specific evolutionary adaptations to pressure that were not previously known, represents the most detailed picture yet of what lives at the bottom of the world. There is one final thing to say about the Mariana Trench, and it is not about the biology or the geology or the history of exploration. It is about the relationship between the trench and time. Deep time, geological time, the kind of time that makes human history seem not just brief, but almost instantaneous. The rock at the western edge of the Pacific Plate, bending downward into the mantle beneath the Mariana Islands at this moment, formed approximately 180 million years ago. The trench itself, in roughly its current form, has existed for perhaps 40 million years. The bacteria living in the sediment at the floor of Challenger Deep are descendants of lineages that stretch back billions of years, and humans have known about the Mariana Trench for approximately 150 years. We have visited its deepest point fewer than a dozen times. We have spent, in total, a few tens of hours at Challenger Deep. The trench does not notice us. The subduction continues at the same rate it has maintained for tens of millions of years. The sediment falls. The microbes process chemical energy in the dark with the same indifference to the surface world that they have always maintained. A visit from a submersible carrying 2 humans lasts, from the trench's perspective, considerably less than a single heartbeat. This could feel diminishing. I don't think it should. The trench's vast indifference to our presence does not reduce the significance of our curiosity about it. If anything, the disproportion is part of what makes the inquiry worthwhile. We're attempting to understand something that operated long before we existed and will continue long after we are gone. This is the most honest description of what science is. The attempt to understand things that do not particularly need to be understood. The Mariana Trench was doing exactly what it does now, while the first mammals scurried between dinosaur legs on the surface above it. It will be doing exactly what it does now, long after everything currently alive has been replaced by whatever comes next. The Pacific Plate will continue to descend until it has been completely consumed into the mantle, a process that will take millions of years, and then the trench will cease to exist in its current form, and whatever comes after us will look at that changed geography and wonder, as we wonder now at the shapes of ancient continents, what the world was like before. We have caught the Mariana Trench in progress. We have looked at it, measured it, sent machines into it and withdrawn them with samples. We have found life where Forbes said there would be none, and plastic bags where no one expected them, and possibly the chemical echoes of life's own beginning in the alkaline venting of its mud volcanoes. We have understood a very small portion of what there is to understand. This is, I think, the most fitting place to end, not with completion, but with the honest acknowledgement that the frontier of knowledge about the deepest place on Earth runs directly through the present moment. The questions the Mariana Trench raises are not historical curiosities. They are open. Let the trench settle into the mind now, cold, dark, immense. Alive in its quiet, persistent, extraordinarily ancient way, 11 kilometres below the surface of the world you are resting in. Somewhere down there, in sediment that has never seen light, in water that has not touched air in thousands of years, life is proceeding with complete indifference to whether we know about it or not. It was there long before we were. It will be there long after. There is something that happens to light below the surface of the ocean that is worth describing with some care, because it shapes everything else about what the trench is. At the surface, on a clear day, Sunlight strikes the water at multiple wavelengths, the full spectrum of visible light plus ultraviolet and infrared. The water begins absorbing that light immediately. Red wavelengths are absorbed first, disappearing within the top few meters. Orange goes next. Yellow follows within 20 or 30 meters. By 50 meters, all that remains of the original sunlight spectrum is blue and green, the wavelengths that water absorbs least efficiently. By 200 meters, even these are gone. Below 200 meters, there is no sunlight at all. This is not gradual dimming, it is total. Below 200 meters, the ocean is as dark as the inside of a sealed container. The darkness is not broken by any natural light source of astronomical origin. No moon, no stars, no aurora, nothing. And yet, the deep ocean is not entirely dark. The creatures that live in the mesopelagic and bathypelagic zones, in the twilight zone and below, produce their own light. This is bioluminescence, and it is not a marginal phenomenon. Estimates suggest that between 76 and 80% of all deep-sea animals produce bioluminescence in some form. The mechanisms vary. Some organisms host symbiotic bacteria that produce light continuously. Others generate light through biochemical reactions within their own cells, using a class of molecules called luciferins, oxidized by enzymes called luciferases, the same basic chemistry that makes a firefly glow on a summer evening, though the specific molecules differ across the evolutionary lineages that have developed it independently. What is bioluminescence for in the deep ocean? The answer is different for different organisms, and the full ecology of deep-sea light is still being studied. Some organisms use light as a lure. The famous anglerfish, with its bioluminescent appendage dangling in front of its mouth, attracting prey drawn to the glow. Some use it as a countershade, producing a faint blue-green luminescence on their ventral surface that matches the residual sunlight filtering down from above, making them effectively transparent to predators looking up from below. Some use it for communication. Patterns of light on their bodies that may function as species recognition signals in darkness, where visual patterns on scales and fins are invisible. Some organisms use bioluminescence defensively. When threatened, they produce a sudden bright flash, the equivalent of a camera strobe, that may temporarily blind a predator or startle it into retreating. Others release clouds of bioluminescent secretions, like a luminescent version of an octopus's ink cloud, that confuse the predator while the prey escapes. The deep-sea ostracod, a tiny crustacean related to a water flea, squirts bioluminescent fluid when swallowed by a fish. The fish, presumably unhappy about glowing from the inside, spits it out. This is life-solving problems. The darkness that would seem to be a pure disadvantage becomes, through evolutionary time, a medium in which entirely new forms of communication and predation and evasion develop. The deep ocean has had hundreds of millions of years to work out the biology of darkness, and what it has produced is extraordinary. Below 4,000 meters, in the abyssal zone and deeper, bioluminescence becomes sparser. The organisms are fewer, the encounters rarer, the ecological pressure for complex signaling somewhat reduced. But it does not disappear entirely. Even at hadal depths, some organisms produce light. Sea cucumbers at the bottom of the trench have been photographed emitting bioluminescent glows when disturbed. Some of the microorganisms in the hadal sediment may produce light as a metabolic byproduct, though this is harder to study. The question of what it would actually be like to descend through the water column of the Mariana Trench, not in a submersible, but as a body, if a body could survive it, is not one that has a scientific answer. But it is worth constructing mentally because it clarifies what the physical reality of the descent actually involves. At the surface, you are in a warm world of light and noise and familiar pressure. The ocean surface, even in the deep Pacific, far from land, is biologically rich. Phytoplankton are visible as colour in the water. Fish and seabirds move through. Sunlight refracts and fragments. You begin to descend. The light dims. Within the first 100 meters, the blue deepens and the red disappears from the spectrum. By the time you reach 50 meters, the world above you is a pale blue-green rectangle of diminishing brightness. By 200 meters, it is gone. You are in darkness so complete that your eyes find nothing to rest on. The temperature drops. In tropical ocean waters, the transition from warm surface water to cold deep water happens between roughly 200 and 1,000 meters, in a zone called the thermocline. It is not a gentle gradient. The temperature can drop by 15 degrees Celsius within a few hundred meters of depth. Below the thermocline, the water stabilizes at between 2 and 4 degrees, and it will maintain that temperature all the way to the floor of the trench. The pressure is accumulating around you with every meter of descent. At 100 meters, it is 11 atmospheres, the pressure that would require a specialized diving suit to survive. At 1,000 meters, 101 atmospheres. At 5,000 meters, 500 atmospheres. This is already beyond any equipment tested by human beings in open water. At 11,000 meters, you arrive at approximately 1,071 atmospheres, or roughly 8 tons pressing on every square inch of your body simultaneously. In the absolute blackness, at near freezing temperature, under this pressure, you touch the floor. What you find there, if you survived it, is sediment, soft, pale, almost lunar in its stillness. No current moves it, no light reaches it. The sound of the surface world, storms, ship engines, whale song, is muffled to near inaudibility by 11 kilometers of water. The floor of the Mariana Trench is one of the quietest places on Earth. And in that quiet, in that cold, in that dark, organisms move. The deep ocean is sometimes described as silent. It is not. Sound travels far better in water than in air, roughly 4 times faster, due to water's greater density and incompressibility, and the deep ocean is full of sources of sound. Earthquakes generate seismic waves that travel through the ocean floor and couple into acoustic signals in the water. Hydrothermal vents produce broadband noise from the rushing of superheated fluid through rock. Marine animals generate sounds across a vast range of frequencies, from the infrasonic rumbles of blue whales at frequencies below 20 Hz, inaudible to human ears, to the high-frequency clicks of sperm whales used for echolocation, which can be heard at ranges of hundreds of kilometers. The deep ocean has a feature of acoustic physics that is among its most remarkable properties. At roughly 600 to 1,000 meters in most parts of the world's oceans, there is a depth of minimum sound velocity, a zone where sound travels more slowly than it does either above or below. This creates a natural waveguide. Sound waves generated in this zone tend to be bent back toward it from both above and below, refracted by the gradients of sound velocity that surround it. The result is that sound injected into this channel, scientists call it the SOFAR channel, for sound fixing and ranging, can travel extraordinary distances with very little energy loss. The United States Navy discovered this property in the 1940s and used it practically. A downed pilot at sea could detonate a small explosive charge at SOFAR depth, and the acoustic signal would travel thousands of kilometers through the channel, detectable at monitoring stations on the far side of the ocean. The Navy used this to rescue pilots until GPS made the system redundant. Whales use the SOFAR channel too. The vocalizations of fin whales and blue whales, produced at frequencies that match the channel's transmission characteristics, may travel half an ocean or more. This is widely accepted among marine biologists, though the full extent and significance of long-range acoustic communication in cetaceans is still being debated. The Mariana Trench lies below the SOFAR channel, and the acoustic environment at the trench floor reflects this. A hydrophone placed at Challenger Deep would detect the geological sounds of the trench itself, the subduction, the occasional seismic events, the chemical venting, filtered through 11 kilometers of overlying water. It would detect some distant sounds transmitted through the SOFAR channel from thousands of kilometers away. In 2016, researchers from NOAA and Oregon State University recorded ambient sounds at Challenger Deep using a specially designed hydrophone, and found a remarkably rich acoustic environment. The trench floor receives the low-frequency rumble of distant earthquakes from across the Pacific. The passage of a large typhoon at the surface was detectable as infrasound nearly 11 kilometers below. Baleen whale vocalizations were present. The bottom of the Mariana Trench is connected to the surface world not only through the slow fall of marine snow and the chemistry of contamination, but through sound. The trench is acoustically coupled to events thousands of kilometers away and thousands of meters above. It is not isolated. It is embedded in the planet's systems in ways that traverse the distance between surface and floor more rapidly and more completely than any physical material could. The organisms of the hadal zone are sometimes described as extremophiles, lovers of extreme conditions, organisms adapted to environments that other life cannot tolerate. This framing is technically accurate, but slightly misleading, because it implies that these organisms experience their environment as extreme. They do not. For a hadal snailfish, the floor of the Mariana Trench is not extreme. It is home. The pressure that would kill a surface fish in seconds is simply the pressure that exists. The darkness that would render a surface fish helpless is simply the environment. What is extreme is a relative judgment that reveals something about the perspective of the observer, not about the conditions experienced by the observed. This is worth pausing on, because it applies more broadly than the deep ocean. Every organism lives in the conditions it evolved in. A tardigrade, the microscopic animal sometimes called a water bear, which can survive exposure to the vacuum of space, temperatures near absolute zero, and radiation levels that would kill a human many times over, does not experience these as extreme. It has the biochemistry to tolerate them. A bacterium living in the superheated fluid of a hydrothermal vent at temperatures above 100 degrees Celsius experiences that temperature as normal. The heat that would denature a mammalian protein is the heat that its enzymes were built for. From this perspective, the deepest life on Earth is not a collection of survivors clinging to existence at the edge of the possible. It is a collection of organisms that are perfectly suited to where they are. The TMAO in a hadal snailfish's tissue is not a desperate improvisation. It is an elegant solution, refined over millions of years to a specific physical challenge. The hadal amphipod's gut microbiome, its pressure-adapted enzymes, its chemical recognition systems for detecting food falls, these are not approximations of a better life that exists at shallower depths. They are optimal for Challenger Deep. We are inclined to see the deep ocean as harsh because we cannot survive there. This is a parochialism of the surface. The deep ocean is not harsh. It is simply different and full of things that are at home in it. The question of what the Mariana Trench means, what it says about the planet, about life, about our place in the natural world, is not a question with a single clean answer, but there are a few things that seem clear after sitting with the evidence carefully. The first is that the boundary between the living and the non-living runs considerably deeper than we once thought. The azoic hypothesis has been definitively refuted, not just for the hadal zone, but for the entire deep ocean and beyond. Microbial life has been found in sediment cores drilled from kilometres beneath the seafloor in rock formations hundreds of millions of years old, in conditions that make the Mariana Trench floor seem almost hospitable by comparison. Life is not a thin veneer on a rock surface illuminated by a nearby star. Life is a deep penetration into any environment where liquid water and chemical energy exist, operating at geological timescales and in geological quantities. The second thing is that the deep ocean is not a passive repository. It is an active participant in the planet's chemistry. The bacteria in hadal sediment process organic carbon and release it as different compounds that affect ocean chemistry. The subduction of the Pacific plate carries surface-derived materials, including the carbon and nitrogen of marine organisms, and now the PCBs and microplastics of industrial civilization, into the mantle, where they are transformed and eventually returned to the surface through volcanism. The hadal zone is a transit point in planetary-scale material cycles that operate on geological timescales. When we contaminate the Mariana Trench with microplastics and persistent organic pollutants and bomb-derived carbon-14, we are not merely spoiling a pristine environment. We are inserting our chemical signature into a planetary cycle. We are changing the inputs to processes that shape ocean chemistry, climate, and the deep carbon cycle. The consequences are not immediate and are not well understood, but they are real. The third thing, the one I find most affecting, is the question of what the depth of ignorance about the ocean means for how we govern our relationship with it. More than 3/4 of the ocean floor remains unmapped. We have directly observed approximately 0.1% of it. We are making decisions about deep-sea mining, about ocean acidification, about deep water fisheries, about the treatment of ocean ecosystems as sinks for our waste, based on knowledge derived from a tiny and geographically biased sample of what the ocean actually contains. This is not a situation unique to the ocean. We are making decisions about the Amazon rainforest based on incomplete taxonomy of its insects and microorganisms. We are making decisions about soil management based on incomplete knowledge of soil microbial ecology. We are constantly acting on partial information with full consequences. But the ocean is a particularly striking case because the scale of what we do not know is so vast, and because the ocean is so central to the functioning of the planetary systems that sustain us. The deep ocean is the largest carbon reservoir in the active Earth system. Changes to its biological productivity, its circulation, its chemistry affect atmospheric carbon, global temperature, and the productivity of surface waters that feed fisheries that feed humans. We are altering this system through warming, through acidification, through physical contamination, through the disruption of deep water circulation patterns, with a degree of understanding that falls well short of what the consequences demand. The Mariana Trench is the extreme case of this. If we know so little about the most studied and most famous deep-sea feature on Earth, what does that imply about everything else? I am not arguing for paralysis. Decisions must be made on incomplete information. The question is whether they are made with appropriate humility, with investment in improving the knowledge base, and with precaution proportionate to the uncertainty. Looking at the current state of deep-sea research funding, the rate of ocean floor mapping, and the trajectory of deep water pollution, the answer is, at best, mixed. Let me say one more thing about the Trieste and about Don Walsh, because the human dimensions of the story deserve attention. Don Walsh volunteered for the Bathyscaphe program in the late 1950s, because hardly anyone else would. The position was not prestigious. Most naval officers regarded it as a dead-end assignment with limited career prospects. Walsh applied because, as he has said multiple times in interviews, he thought it would be fun. On the morning of the 23rd of January, 1960, before the dive, the weather was bad. Heavy swells, overcast sky, the Trieste rolling badly at the surface. The preparation had been difficult. The tow from Guam had damaged the vessel. The support ships were struggling in the sea conditions. Jacques Piccard recorded in his notes that conditions were discouraging and that the situation looked dangerous. Walsh climbed aboard the pitching Trieste and assessed the same conditions and agreed to descend. The crack at 9,000 meters, the 20 minutes of visibility zero at the bottom, the careful decisions made in the moment with incomplete information. Walsh has said that he and Piccard discussed the crack, assessed the risk as acceptable given that there was no pressure breach, and continued, "This is not recklessness. It is a calibrated judgment made by two people who had thought carefully about the worst case and concluded that the structure was still sound." When they surfaced, Walsh expected that the next team would follow within 2 or 3 years. He was wrong by half a century. He has expressed this disappointment openly and without bitterness. The Navy made decisions based on budget and priority, not on the scientific value of the dives. The Trieste was too expensive to operate and too limited scientifically for the data it returned. The program was wound down. Walsh went on to a long naval career and eventually a PhD in ocean engineering, and spent decades as a voice for deep ocean exploration. In 2019, when Victor Vescovo made his record dives to Challenger Deep, Walsh was invited on the expedition. He was 87 years old. He did not dive to the bottom again. The limiting factor sphere was not large enough for 2 people, but he was present on the surface ship, part of the team. He watched the vehicle descend. He had waited 59 years for someone to return. "I salute Victor Vescovo and his outstanding team," Walsh said publicly after the dive. "Now in the winter of my life, it was a great honour to be invited on this expedition to a place of my youth. A place of my youth, the bottom of the world, 11 kilometres down." There is something in that phrase that stays with me. Not the sentiment of it, though that is real, but the framing. The Challenger Deep is a place. It is somewhere specific, with geography and biological character and physical properties. Walsh went there as a young man and spent 20 minutes in the dark with zero visibility, and came back to the surface and spent the rest of his life thinking about it. He thinks of it the way someone thinks of a place they went to once and have carried with them ever since. There are fewer than a dozen people alive who can say they have been to Challenger Deep. This is not because it is particularly dangerous, though it is not easy, but because the apparatus required to get there is so expensive and specialised that very few organisations have built or funded it. The barrier is mostly economic, not physical or scientific. This may change over the next few decades as submersible technology improves, as the cost of deep-sea vehicles decreases, and as the geopolitical value of deep-sea access becomes clearer. Whether expanded access to the hadal zone will improve our knowledge faster than the ecosystem can be damaged by expanding pollution and climate change is an open question. It should not be. The last light in the ocean disappears at roughly 200 meters depth, depending on water clarity. This is a fact that is easy to state and surprisingly difficult to absorb. There is no light below 200 meters, not dimness, not weak light that could theoretically be perceived by a very sensitive eye. No light at all, no photons of the wavelengths detectable by biological visual systems, for the remaining 10,700 meters of the water column above Challenger Deep. The visual world that organizes so much of our experience of reality simply does not exist there. We are deeply visual animals. Our language is saturated with visual metaphors. We speak of clarity and obscurity, of enlightenment and being in the dark, of seeing and understanding as essentially the same act. The majority of our cerebral cortex is devoted to processing visual information. When we try to imagine a world without light, we tend to imagine darkness as the absence of something, as deprivation. But the organisms of the deep ocean did not evolve in the light and then lose it. They evolved in darkness, or their ancestors did, over the hundreds of millions of years since the deep ocean became inhabited. For them, darkness is not the absence of light. It is just the world. Their senses, pressure reception, chemoreception, mechanoreception, whatever bioluminescent visual capacity some of them have, are tuned to what exists, not to what we experience as normal. There is a philosophical point here that extends beyond biology. The category of extremity is observer-dependent. What is extreme is always extreme relative to something else, to some standard of normality that is itself contingent. For a bacterium in a hydrothermal vent at 120 degrees Celsius, room temperature is extreme. It is far below the organism's functional range. For a surface fish, the Mariana Trench is extreme. For a Mariana snailfish, the surface ocean is lethal. The Mariana Trench is extreme from our perspective. From its own inhabitants' perspective, it is ordinary. This distinction matters because it affects how we think about the trench and about the broader question of life's limits. If we ask, how does life survive in such extreme conditions, we are framing the question from the outside. If we ask, what does it mean that life has evolved to occupy this specific set of conditions, we are framing it from the inside, from the perspective of the evolutionary history of the organisms themselves. The second framing is more honest and more instructive. What it means is that life explores. Over geological time, it fills available space. It exploits available energy. It adapts to whatever physical conditions persist long enough for evolution to find a solution. The hadal zone has existed for tens of millions of years. That is an enormous amount of time for evolution to work. The organisms there are not struggling at the edge of the possible. They are exactly where hundreds of millions of years of natural selection placed them. The serpentinite mud volcanoes of the Mariana Forearc do something that is, by any measure, extraordinary. Serpentinization is a chemical reaction between olivine and pyroxene, minerals in the upper mantle, and water. When oceanic crust is subducted beneath the Mariana Plate and descends into the mantle, it carries seawater in its pores and in the hydrated minerals of its altered upper layers. As it descends and heats, this water is released, what geologists call slab dehydration, and it rises into the overlying mantle, reacting with the mantle peridotite. The reactions produce serpentinite, a slippery, greenish mineral that is physically weaker than the original rock and provides a pathway for fluids and the occasional fragment of mantle material to rise toward the surface. At the Mariana Forearc, this process has built enormous mud volcanoes, structures up to 31 kilometres in diameter and over a kilometre high, dwarfing anything produced by conventional volcanism. These are not mountains of lava. They are mountains of chemically transformed mantle material, extruded slowly over millions of years, fed by the fluid and material released from the subducting Pacific Plate below. The fluids venting from these mud volcanoes are extraordinary in their chemistry. Some have pH values approaching 12, highly alkaline, analogous to industrial lye. They carry dissolved hydrogen gas, methane, and other reduced compounds. The temperatures of these fluids are low by hydrothermal standards, typically below 40 degrees Celsius. But the chemistry makes them highly reactive and surprisingly capable of supporting biological communities. Among the materials erupted from these mud volcanoes are fragments of the subducting Pacific plate itself, pieces of ancient basaltic crust, fragments of seamounts that were riding on the Pacific plate and got caught in the subduction, and even, as has been documented by researchers including Patricia Fryer of the University of Hawaii, intact sections of Cretaceous coral reef. These sections of reef have been carried down to the subduction zone on the Pacific plate, partially subducted, and then brought back to near surface levels by the mud volcano eruption, emerging at the seafloor as coherent sections of ancient limestone, with coral fossils still visible in the rock. Coral from the Cretaceous, 110 million years old approximately, carried down into the subduction zone on a plate of rock moving a few centimeters per year, subjected to the chemistry of the deep earth, and then extruded back toward the surface in a plume of alkaline serpentinite mud. The planet recycles with a thoroughness that has no human parallel. The organic matter detected in samples from these mud volcanoes by researchers from the Massachusetts Institute of Technology and other institutions in the early 2010s has a molecular signature consistent with microbial activity deep within the volcano's plumbing system, not just at the seafloor vent. Deep inside, in conditions of heat and pressure and chemistry that are extraordinary by any measure. The implication is that microbial life may be extending into the mud volcano system kilometres below the seafloor, accessing chemical energy from the serpentinization reactions that power the system from below. This is relevant to the origin of life question in a specific way. The alkaline, hydrogen-rich conditions in serpentinizing systems have been proposed as the environment in which the first self-sustaining biochemistry might have arisen on early Earth, before photosynthesis, before complex molecules, before the first cell membrane. The serpentinite mud volcanoes of the Mariana Forearc are not identical to the proposed early Earth conditions, but they represent a class of geological environment that modern researchers believe may have been widespread on the young Earth and that may currently exist on Enceladus and potentially Europa. If the origin of life occurred in a serpentinizing alkaline vent, and this remains a hypothesis, not a consensus, then the serpentinite chemistry of the Mariana Forearc is not merely geologically interesting. It is a potential window into the conditions of life's own beginning. One thing about the Mariana Trench that I have not yet addressed directly is its size, not its depth, but its horizontal extent. The Challenger Deep, which most discussions focus on, is a relatively small feature within the larger trench system. It occupies a slot-shaped valley approximately 11 kilometres long and roughly 1.5 kilometres wide at its deepest section, at the southern end of the trench. The Sirena Deep, lying about 240 kilometres to the east, reaches nearly 10.5 kilometres depth and is actually the second deepest point confirmed in the Mariana Trench. The entire trench system is approximately 2,550 kilometres long and up to 69 kilometres wide. Most of this has never been visited by any vehicle. The multi-beam sonar surveys that have been conducted give a topographic picture of the trench's floor. But sonar maps are not observations. They tell you the shape of things. They do not tell you what is living there, what the sediment chemistry is, what the microbial communities are doing, or whether there are geological features, cold seeps, small hydrothermal systems, unusual rock exposures that would be scientifically valuable. A 2025 NOAA Ocean Exploration Expedition planned to systematically survey areas of the Mariana Trench Marine National Monument that had never been visited, using remotely operated vehicles and mapping systems. The majority of the monument, despite covering approximately 246,000 square kilometres, had not been explored with modern technologies at the time the expedition was organised. The water column above the trench floor, 11 kilometres of biology and chemistry, was described by the expedition planners as virtually unexplored. The trench's entire water column is a research frontier. The deep mid-water region, the 3 to 9 kilometre depth range, is particularly poorly studied because it sits between the focus of surface biology above and the hadal floor below. The organisms that live in this intermediate zone, the processes that occur there, the chemical gradients that exist through it, all of these are poorly characterized. Some of the most scientifically valuable discoveries of the next few decades will likely come from systematic study of exactly this region. The trench is quiet now. as it was quiet before the first human ever crossed the Pacific Ocean above it, as it will be quiet long after the last. The sediment accumulates at approximately 1 to 2 millimeters per 1,000 years at the floor of Challenger Deep. It has been doing this since before the trench existed in its current form, and it will continue doing it long after anyone now alive is gone. Each millimeter of sediment is 1,000 years of marine snow compressed and preserved, a record of surface productivity, ocean chemistry, climate, and the biological world above. Scientists drill sediment cores and read them like books. The chemical signatures preserved in them, isotope ratios, molecular fossils of ancient organisms, the mineral records of ocean temperature and circulation, tell the story of the ocean's past with a precision that would have seemed miraculous to any previous generation of researchers. The sediment at the floor of the Mariana Trench has been accumulating this record for tens of millions of years. We have sampled a very small portion of it. Everything we know about the Mariana Trench, its depth, its geology, its biology, its contamination, its potential relevance to astrobiology, and to the origin of life, comes from a small number of expeditions, each lasting a few weeks, each visiting a few specific locations, each collecting samples from a small area of a vast and ancient system. This is not a failure of science. This is science at an early stage. The Mariana Trench has been seriously studied for less than 50 years, and with genuinely capable technology for less than 30. What has been learned in that time is extraordinary. What remains to be learned is, almost certainly, more extraordinary still. The trench waits. It has always waited. It is extraordinarily patient, in the way that geology is patient, in the way that deep time is patient. It will still be there when better vehicles descend, when better sensors are deployed, when the science of deep ocean biology and chemistry has advanced to the point where we can begin to understand what we are actually looking at. It offers no resistance to being known. It simply requires us to come to it on its own terms, slowly, carefully, with enormous investment and genuine curiosity and the willingness to be surprised. When you lie in darkness, as you are now, there is a similarity to the trench floor that is easy to overlook. The dark is the same dark. The temperature difference is enormous, but the quality of the darkness, the complete absence of light, is identical. The silence is the same silence, or something close to it, below the noise of the surface world, in still air or still water. The background of the universe is quiet. What the trench has, that the room you are resting in does not, is depth. Not metaphorical depth, literal depth. 11 kilometres of water overhead, pressing down with the weight of the entire ocean column. The organisms at the bottom of the Mariana Trench have the entire ocean above them. They are at the bottom of the world's largest structure. And yet they persist. They do what they have always done. They process energy, they reproduce, they die, and their material is incorporated into the sediment or consumed by the organisms that come after them. The cycle continues without pause, without awareness of the surface world, without any orientation toward anything we would recognize as purpose. There is a stillness in that image that is worth carrying into sleep. The deepest place on earth is cold and dark and full of life, going about its business with perfect indifference to whether it is observed. It was there before we were. It will be there after. The ocean above it is 11 kilometres of cold water, pressing down gently, filtering out every photon, holding everything still. Let the weight of that settle in the body now. Not the pressure, the stillness, the deep permanence of a place that has been exactly itself for longer than we can imagine, that asks nothing of us and receives nothing we intend to give it, that is simply there, in the dark, at the bottom of the world. Sleep now. The trench is patient. The ocean is quiet. The night is long. Something that is rarely discussed in accounts of the Mariana Trench is the sheer practical difficulty of measuring it accurately. The depth of Challenger Deep has been measured many times, and each measurement has produced a slightly different number. The 1875 HMS Challenger sounding recorded approximately 8,184 metres. The 1951 HMS Challenger 2 measurement came in around 10,863 metres. The 1960 Trieste dive registered 11,521 metres on its instruments, a figure later revised sharply downward as more precise measurements became available. James Cameron's dive in 2012 recorded 10,908 metres. Victor Vescovo's 2019 dive placed the record at 10,927 metres. A 2021 study using submersible-mounted pressure sensors and acoustic positioning, published in the journal Deep Sea Research, revised the canonical depth to 10,935 metres, with an uncertainty of approximately 6 metres. Why is measuring the depth of the ocean so difficult? The first challenge is that depth measurement in the open ocean cannot be done with a ruler. The traditional method, lowering a weighted line and feeling for the bottom, is imprecise, because the line drifts with ocean currents during the descent, making the measured distance greater than the true vertical depth. Modern methods use acoustic sonar. A sound pulse is transmitted downward, bounces off the seafloor, and returns. The time elapsed divided by the speed of sound in water gives the depth. But the speed of sound in water is not constant. It varies with temperature, salinity, and pressure, all three of which change substantially across 11 kilometres of water column. To calculate depth accurately from a sound travel time, you need to know the sound velocity profile of the entire water column along the path of the pulse. This requires measurements of temperature, salinity, and pressure at multiple depths during the same dive, which is expensive and technically demanding. Additionally, the Challenger Deep is not a single point. It has three distinct basins: the Eastern Pool, the Central Pool, and the Western Pool, each with slightly different maximum depths. Early measurements that located the deepest point slightly differently from later ones were not necessarily wrong. They may have been measuring different subfeatures within the broader Challenger Deep structure. The current best measurement, placing the maximum depth of the Eastern Pool at 10,935 metres with an uncertainty of 6 metres, represents the most rigorous application of modern pressure sensor technology and acoustic positioning. Whether future measurements with improved equipment will revise this further is impossible to predict. The basic uncertainty of a few meters has been remarkably persistent across different measurement approaches. This irreducible measurement uncertainty is itself a kind of lesson. The deepest place on Earth, a geographical feature of enormous significance, the subject of major expeditions and considerable scientific investment, remains known to within a few meters, but not with absolute precision. We know approximately where it is and approximately how deep it is. We have not nailed it down with the certainty we might expect. The ocean does not yield easily to measurement. It is a dynamic, three-dimensional, variable medium in which the very properties that allow us to probe it, sound velocity, water transparency, electronic transmission, change with depth in ways that introduce uncertainty. This is not a failure of instrumentation. It is a property of the system being measured. The oldest material that will ever reach the floor of the Mariana Trench has already been there for a very long time. The sediment at Challenger Deep contains, among its components, the skeletal remains of foraminifera, single-celled organisms with elaborate mineral shells that have been falling through the water column and accumulating on the ocean floor for hundreds of millions of years. These shells are preserved in the sediment after the organisms die, providing a record of past ocean conditions, temperature, chemistry, productivity, that geochemists can read from the changing ratios of isotopes in the carbonate minerals. Some of the foraminifera, preserved in deep-sea sediment, belong to lineages that were already old when the dinosaurs appeared. The group as a whole extends back over 500 million years, to the Cambrian period, when multicellular animal life was just beginning to diversify in the Earth's oceans. The deep-sea sediment record is, in this sense, a continuous archive of the history of life in the ocean, imperfect, incomplete, but extending back further than almost any other geological record available. The sediment at the floor of the Mariana Trench contains this archive. It contains the mineralized remains of organisms that lived during the age of dinosaurs, during the age of giant marine reptiles, during the great mass extinctions that have punctuated the history of animal life. It contains signatures of climate events, ancient ice ages, ancient warm periods, ancient volcanic crises, preserved in the chemistry of tiny shells that fell through the water column and settled on the bottom, undisturbed in the extreme cold and dark. Drilling into this sediment, recovering cores that extend tens of meters below the modern floor, would be an extraordinary scientific undertaking. The technology to drill at full hadal depth does not yet exist in operational form. though research programs are exploring the possibility. A sediment core from Challenger Deep, extending several million years back in time, would contain a climate and biological record for the deep Pacific that would be unlike any other archive available. The sediment waits. The Mariana Trench is surrounded by a political and legal framework that reflects its unique status. In 2009, the United States government designated the Marianas Trench Marine National Monument under the Antiquities Act, protecting approximately 246,000 square kilometres of submerged land around the Mariana Islands from extractive uses, including fishing and mining. The monument includes the trench itself, along with the island unit protecting the waters around 3 of the northern Mariana Islands, and the volcanic unit protecting the submarine volcanic arc and associated features to the north. The creation of the monument reflected growing scientific understanding of the trench's biological and geological importance, and the interest of the government of the Commonwealth of the Northern Mariana Islands in protecting the region's unique ecosystems. It provides a degree of legal protection that is relatively rare for deep-sea features. The broader question of deep-sea governance outside national jurisdiction, the so-called high seas, which cover approximately 60% of the ocean's surface and a comparable proportion of the deep ocean floor, has been one of the most difficult and contested areas of international environmental law for decades. A high seas treaty negotiated under the United Nations Convention on the Law of the Sea was agreed in principle in 2023, providing a framework for the protection of marine biodiversity in areas beyond national jurisdiction. The implementation of this treaty, what it will actually mean in practice for deep-sea habitats, deep-sea mining regulation, and the protection of hadal ecosystems, remains to be worked out. The economic interests at stake are significant. Deep-sea mineral resources, polymetallic nodules, cobalt-rich ferromanganese crusts, seafloor massive sulfide deposits, represent enormous quantities of minerals needed for the green energy transition. The International Seabed Authority, which regulates mineral extraction in international waters under the Law of the Sea Convention, has been developing a mining code for decades, repeatedly delayed by disagreement between states that favour rapid development of these resources and those that favour stronger environmental protection. The Mariana Trench, as a nationally protected monument, is largely outside this particular dispute. But the broader principles, how much we know about deep-sea ecosystems, what the consequences of disturbing them are, whether recovery is possible, and on what timescales, apply to the trench as much as to any other deep-sea feature. The science of deep-sea ecology is not sufficiently advanced to answer these questions with confidence. This is not a reason to avoid making decisions. It is a reason to make them with more humility and more precaution than the economic pressures alone would encourage. There are aspects of the Mariana Trench that we will probably never fully understand. Some of these are practical limitations. The bottom sediment of Challenger Deep, for instance, contains a biological and chemical record extending millions of years back in time. To read that record fully, you would need sediment cores extending tens of meters below the modern surface. The pressure at Challenger Deep makes conventional drilling equipment extremely difficult to operate. No sediment cores have been successfully recovered from the deepest parts of the trench, and the technology to do so at scale does not yet exist. The record is there, we cannot yet access it. Some limitations are more fundamental. The deep-sea biological communities that have been discovered at various hadal sites are characterized from small numbers of samples taken at specific times. The temporal variability of these communities, how they respond to seasonal changes in surface productivity, to episodic food falls, to seismic events, to the long-term trends in ocean chemistry driven by climate change, is almost completely unknown. We have snapshots. We do not have the longitudinal record needed to understand dynamics, and some of what we do not know may be permanently unknowable in a meaningful sense. The evolutionary history of hadal organisms, how the lineages now occupying the trench originally colonized it, what the trench community looked like 40 million years ago when the trench first formed, how the isolation of different trench populations has proceeded over geological time. These questions are approachable through genomics and fossil records only to a limited extent. The deep-sea sediment record contains biological information, but not detailed enough to reconstruct the ecological history of specific trench communities. What we know about the Mariana Trench is significant. What we know is real and hard-won and sometimes beautiful. The discovery of the TMAO depth gradient is elegant, a clean experimental relationship between a chemical adaptation and the environment that selected for it. The discovery of chemosynthetic microbial communities at extreme depth is genuinely transformative for our understanding of life's range. The accumulation of contamination data, the plastics, the PCBs, the bomb carbon, is sobering and important. But the ratio of what is known to what might be known is very small. The trench is a vast system, ancient and complex and barely studied. What understanding we have of it is a beginning, not a conclusion. There is something specific that happens in the mind when you contemplate the Mariana Trench seriously. Not just noting its statistics, but actually trying to hold it in the imagination as a real place that exists right now, while you are awake or asleep, while you are doing whatever else you do in your life. It is one of those facts that keeps not quite landing, 11 kilometres deep. You accept it, and then a few minutes later you find yourself accepting it again, as if it didn't quite take the first time. The depth is so far outside the range of direct human experience that it resists mental representation. We don't have a frame of reference that makes 11 kilometres of water feel like a real distance rather than an abstraction. The analogies, it's deeper than Everest, is tall, it's 11 times the height of a skyscraper, it's 7 miles down, help some, but they can only gesture at the thing, not produce it in the mind. The distance between the surface and the floor of Challenger Deep is genuinely difficult to hold as an image. What sometimes helps is time instead of distance. The descent in the Trieste took 4 hours and 47 minutes. That is a span of time the mind can hold. Nearly 5 hours of falling through darkness and cold and increasing pressure, watching the instruments, the world getting further away and the floor getting closer. Nearly 5 hours to cover 11 kilometres, and in those nearly 5 hours, if you were inside the sphere, there would be nothing to see. Total darkness outside the viewport, except for the occasional bioluminescent flash of an organism encountering the Trieste as it fell, the creak of the hull under increasing pressure, the temperature dropping, the sound of the ballast systems and the life support, the instruments. What Piccard and Walsh thought about during those 5 hours is not something that has been extensively recorded. Walsh has been matter-of-fact about the experience in interviews, focused on the technical aspects, the monitoring of instruments, the management of the dive. Piccard, who died in 2008, wrote about the dive in his 1961 book, 7 Miles Down, but his account was also largely technical. Neither man made particularly dramatic claims about the inner experience of the descent. This restraint seems right to me. The dive was not primarily an emotional event. It was a technical accomplishment undertaken by two people with high levels of training and genuine investment in the outcome. The fact that it was also historically extraordinary was something they understood, but perhaps chose to frame in terms of the work rather than the feeling. What the trench asked of them was attention and competence. They gave it both. The result was 20 minutes at the bottom of the world, a cloud of disturbed ooze, a possibly misidentified flatfish, and the beginning of a field of knowledge that is still being built 65 years later. When we sleep, the body reduces its metabolic rate, lowers its temperature slightly, decreases its oxygen consumption, and withdraws much of its awareness from the external world. The analogy to deep-sea organisms is imperfect, but not entirely without substance. Deep-sea organisms at hadal depths also have reduced metabolic rates, dramatically reduced compared to comparable animals at shallower depths, as an adaptation to the sparse food supply and the physical challenges of extreme pressure. They have slowed their biochemistry to match the pace of the environment. Sleep is not, of course, a metabolic adaptation to food scarcity, but the deepening of rest, the letting go of the need to process and respond to the world, shares something with the deep ocean's stillness. Both involve a reduction in the rate at which the world imposes itself on the interior. The floor of the Mariana Trench is, in this sense, a sustained version of what sleep aspires to be. Still, cold, dark, metabolically reduced, operating on longer timescales than the surface world, persistent across vast spans of time with small, slow changes that add up only over centuries and millennia. There is comfort in that image if you let it be comfortable rather than forbidding. The deepest place on Earth is not empty or desolate. It is alive, quietly and persistently, with organisms going about their business in the same way they have for millions of years. It is patient, it is ancient, it is exactly what it is, without apology or drama. 11 kilometres of ocean above a cold, still floor, where life persists in the dark, with complete indifference to being observed. As it has always done, as it will continue to do, long after the surface world has changed in ways none of us can predict. There is one aspect of deep ocean biology that receives less attention than the dramatic megafauna, the snailfish, the amphipods, the xenophyophores, but may turn out to be scientifically more significant. It is the biology of the sediment itself. The top few centimetres of sediment on the floor of the Mariana Trench are biologically active in a way that is difficult to observe directly, but has been demonstrated through chemical measurements and the analysis of sediment cores. Microbial communities process organic carbon arriving as marine snow, breaking down complex molecules and releasing nutrients back into the water column. Myofauna, tiny organisms in the size range between 1/10 of a millimetre and 1 millimetre, burrow through the sediment, grazing on bacteria and redistributing organic material. A 2013 study published in Nature Geoscience found high rates of microbial carbon turnover in sediment from the Challenger Deep. The rate at which organic carbon is being processed by microbial communities that were substantially higher than in comparable abyssal sediment outside the trench. The authors proposed that the concentration of organic material in the trench by its funnel geometry, combined with the pressure-adapted biochemistry of the microbial communities, produces a system that is more biologically active than its extreme conditions might suggest. This has implications for the global carbon cycle. The deep ocean sediment is an important sink for organic carbon, material that arrives from the surface and is processed, buried, and ultimately preserved in geological deposits. The rate at which this carbon is processed versus preserved determines how much carbon is removed from the active ocean atmosphere system and sequestered in geological timescales. If trench sediments are unusually active in processing organic carbon, they may play a disproportionate role in the global carbon cycle relative to their small total area. The Mariana Trench covers roughly 500,000 square kilometres of seafloor, a small fraction of the total ocean floor. But if the biological activity in that area is substantially higher per unit of seafloor than in comparable abyssal plains, the trench's contribution to deep ocean carbon processing could be significant. This is speculative and requires better data to evaluate, but it illustrates how apparently isolated extreme environments can connect to global-scale processes through the chemistry they perform. The Pacific Plate, as it descends beneath the Mariana Islands, carries something remarkable: traces of the atmosphere and ocean from when the plate was at the surface, tens of millions of years ago. The sediment that accumulates on oceanic crust as it forms and moves across the ocean basin records the composition of the ocean and atmosphere above it. Organic material that settles into the sediment carries isotopic signatures of atmospheric carbon dioxide. Marine organisms whose shells are preserved in the sediment record ocean temperature and chemistry through their mineral composition. When this crust is subducted, these materials are carried down into the mantle, where they are transformed by heat and pressure and eventually recycled back into the Earth's interior. Some fraction of this material is not fully processed. It rises with the slab-derived fluids that serpentinize the overlying mantle and emerge at the mud volcanoes of the Mariana Forearc. The organic molecules detected in samples from these mud volcanoes, whose origin is still debated, may include material that was in the ocean or on the seafloor tens of millions of years ago, preserved through the subduction process and now re-emerging at the surface. The Earth breathes in slow cycles, drawing down the products of surface life and eventually returning them transformed. The Mariana Trench is a visible feature of one of these cycles. The subduction of oceanic crust and the mud volcanoes of the Forearc are among the exhaust vents of the cycle's return path. To sit with this image is to appreciate the planet as a system rather than a collection of features. The trench is not separate from the rest of the world. It is embedded in planetary scale processes of extraordinary reach and duration. The same processes that built the Andes and the Himalayas, that regulated the composition of Earth's atmosphere over geological time, that drove the mass extinctions and the recoveries that followed. These processes include the subduction that created the Mariana Trench and sustains it. The trench is a seam where the planet shows its working, where the mechanism becomes briefly visible, where you can, if you know what to look for, read the slow writing of the Earth's interior against the floor of the ocean. Every civilization that has ever flourished on the surface of the Earth has done so above an ocean that was already ancient, already deep, already full of life in its darkest and most inaccessible places. The Romans and the Chinese and the Egyptians and the Maya and every other human culture that left a record behind, none of them knew the Mariana Trench existed. They sailed on an ocean whose floor was completely unknown to them. The trench was there. It has always been there. It was there when the first anatomically modern humans left Africa. It was there when the ancestors of the Mariana Islands' first inhabitants crossed the Pacific in canoes, navigating by stars and wave patterns across an ocean they had never crossed before. It was there, silent and dark and 11 kilometres deep, while all of recorded human history unfolded on the surface above it. The knowledge we now have of the trench, imperfect, partial, recently acquired, does not change what the trench is or has been. It changes only our relationship to it. We are, very recently, aware of a feature of the planet that was completely unknown to every human generation before us. We have sent vehicles to its bottom and retrieved samples and learned something, a small amount, of what it contains. In another 100 years, if the science of deep ocean exploration advances as it has over the past 100, the picture will be dramatically richer. The trench that our descendants will understand will be continuous with the trench we glimpse now. Same geology, same organisms, same place, but known with a depth and detail that is currently not imaginable. Or it will be changed. Warmer ocean water is already penetrating into deep sea environments at rates that were not predicted even a decade ago. The chemistry of the deep ocean is shifting in response to increased atmospheric carbon dioxide. The contamination of hadal environments with persistent pollutants is ongoing and accumulating. Whether the Mariana Trench of 200 years from now will be recognizably similar to the trench of today, or whether it will have been altered by a century of accelerating human impact, is a question that cannot be answered yet. What we do now, with the knowledge we have, will shape the answer. This is not uniquely true of the Mariana Trench. It is true of everything. But the trench is a particularly instructive case because it is simultaneously the most extreme and inaccessible environment on Earth, and one of the places most demonstrably affected by the accumulated byproducts of surface human activity. Plastic from a factory somewhere in Asia, chemicals from electrical equipment banned 40 years ago, radiocarbon from atomic tests in the Nevada desert. All of it finds its way, eventually, to the bottom of the world. The trench asks nothing of us, but it receives everything we send.